Thanks again, Caitlin, for stepping up to that. Caitlin received her bachelor's from Harvard, where she studied environmental science and public policy, her master's of science in ecological planning from the University of Vermont, and her PhD from BU, where she studied under Richard Primack in studying biology, and did a floristic and phenological review of the New England region over the past 100 years, and she'll be presenting on some current research which delves a bit deeper in time.
I would just add that review was featured in Redora last spring. So and one of the featured reviews in Redora
received quite a bit of publicity as well.
Yeah.
Welcome, Caitlin Douglas MacKenzie.
Thank you so much. All right, thank you. I am really excited to be speaking to everyone, and hopefully, even though I don't have a lot of final results to share, you'll still get a sense of what happens in Jacqueline's wonderful lab at University of Maine, where I've been a postdoc for the last 2 years. Yeah. All right, but I just wanted to start by acknowledging that the landscape where we work in Acadia National Park is part of the Wabanaki territory, and they have been on this landscape for as long as my paleoecological study. So to start, there's just not a lot of alpine habitat east of the Mississippi. This map shows pretty much all of it. If you put it all together, you could fit it inside the city of Boston 4 times. And so we have very small, scattered, isolated populations of alpine and subalpine plant communities. And our mountains are comparatively fun-sized. So if you lined up Cadillac Mountain next to Mount Princeton in Colorado, we get dwarfed. And in fact, in the state of Colorado, the lowest point on the landscape is still taller than Mount Washington, the tallest point in the Northeast. So you literally cannot get to the top of Mount Washington if you are in Colorado. And so that means that if you are an alpine plant living in New England, you don't have a lot of space upslope because there isn't very much upslope to be had. But these really small, isolated alpine systems have really long histories of conservation in the Northeast. So the Adirondacks, the Green Mountains, the White Mountains, the Appalachian Mountains, Acadia National Park, Baxter State Park, these are all kind of the foundational conservation stories about protected landscapes in the Northeast, and many of them have been protected for over a century now. And so we have these really wonderful alpine and subalpine systems that protect these beautiful plant communities. But if we're looking ahead, we know that our conservation landscape doesn't protect these species from climate change. And so managers who have now been working on these landscapes for about a century are faced with a new challenge of how do you manage a system that doesn't have very much room to move upslope and can't really migrate north without going across a totally different landscape. And in general, these montane and alpine systems are just seen as these harbingers of climate change. So most alpine systems, most Arctic systems are considered to be better barometers of climate change than microrefugia from it. And the IPCC has said over and over again that they have really high confidence that these alpine systems are already showing high sensitivity to climate change, and they'll be vulnerable to it in the future. And in fact, they talk specifically about tree line and how tree line is already changing, but it's unclear how climate change is driving that, and especially as you move from location to location. And in fact, the IPCC is not the first body to consider the role of climate change and tree line. Back in the 1940s, the Ecological Society of America had a meeting on top of Mount Washington, and I don't really know the logistical details of how they did that, but this address from this guy named Robert Griggs talked about how we should look at the Arctic plants here on Mount Washington, hanging on at these trees, stretching up the mountain slopes as mute indicators of climatic trends, and we should seek to unravel their story. And part of his address actually talked about the importance of alpine plants in our general understanding of Darwin's theory of evolution, because before Darwin, these disjunct populations were considered to be evidence that creation had to happen multiple times. So if you consider diapensia and its kind of circumarctic, circumboreal distribution, these naturalists, I guess, were under the impression that God was like, all right, so we're going to have diapensia here, and here, and here, and here, and here, and each time there was a diapensia population, it was like a different origin story. And so what the kind of giants of the New England botanical scene had done for Darwin, including people like Asa Gray, was understand that these populations actually represented, they thought, these kind of alpine relics from glacial, from the glacial history of the Northeast. So as glaciers came in, all of these Arctic species had kind of migrated south, and then glaciers leave, and they migrate north, and they end up on these kind of disjunct populations. And that's a great story. And if we look at the last 70 years since that talk on top of Mount Washington, there's evidence that the summit of Mount Washington is actually maybe one of those microrefugia. It's warming on top of Mount Washington at the observatory station, but it's not statistically warmer than it used to be, and that is very different from the temperature records at the bottom of Mount Washington. And in fact, we have the same understanding of what's happening on smaller mountains like Cadillac Mountain and Acadia National Park. When I worked on my dissertation research on Cadillac, Sargent, and Pematic Mountains, we put out temperature loggers across elevation zones and aspects. And what we found was that microclimate and elevation are decoupled on these mountains. And so there is evidence that the tops of these mountains, these alpine and subalpine habitats, are warming differently than the rest of the region and the globe. And a pretty large climate change vulnerability assessment that came out from the state of Maine in 2014 basically said that our alpine and mountain key habitats are really highly vulnerable, and they're vulnerable because the area that we have that's above tree line is predicted to decline. But even with that, we'll likely still have some alpine persistence in the state of Maine. And I thought this was an interesting finding around climate change vulnerability, because I had spent my whole dissertation looking at climate change vulnerability for subalpine plants in Acadia National Park. And so when I dug into this a little bit more, there were two citations that came up. So alpine habitat islands smaller than Mount Katahdin may be lost. And Kimball, 1997. So Kimball was my boss when I worked at the Appalachian Mountain Club. And he's a great guy, but this is a white paper with back of the envelope calculations. And I don't think that he meant it to be used in a way that would then tell us like whether we could expect to see alpine plants on top of Mount Katahdin based on calculations of like what was happening in the European Alps. And then the persistence of alpine communities during a warming period 5,000 years ago suggests that many alpine plant habitats may persist through 2100. And so that Miller and Spear paper is from Mount Washington. They cored lakes of the clouds on Mount Washington and found evidence in the form of fossilized achenes from Pontilla plants that suggests that that habitat was open throughout the Holocene. But they readily admit in that paper that they did not look at a fine enough temporal resolution to really capture vegetation changes to rapid warming events. And so we have a back of the envelope calculation, and we have a core from Mount Washington trying to tell us what we can expect in the state of Maine for our alpine plant populations. And I felt like that was not enough information. And I felt that especially because I had gone to Jacqueline Gill as a neo-ecologist at the end of my PhD. I'd been working on like forest exchange over the course of the 20th century. And working in Acadia with these low mountains, hiking around above tree line all the time, and just kind of wondering why we had these subalpine plant communities at 1,500 feet. And I went to Jacqueline and I said, you are a paleoecologist. Are these communities on top of Cadillac Mountain post-glacial tundra relics? And Jacqueline said, well, they're supposed to be, but we actually don't know because we don't have a lot of paleo data from areas that are above tree line. And so we can see why places like that climate change vulnerability assessment for the state of Maine was relying on a calculation from a white paper and a core from Mount Washington to come up with the paleo history of what they could expect in Maine. And when I talk about how there's not a lot of cores from above tree line, that's in really great contrast to what's happening across the rest of New England. Lowland areas in New England that have glacial kettle holes are just really well documented in the paleo record. We core those things all the time. This is just one paper that was able to pull all these different cores together. All of the triangles, squares, crosses, circles, those are all different ponds that have been cored, and they're pollen records put together to look at past vegetation changes. And so when you pull up one of these sediment cores and then look at the pollen that's captured in those layers, and if you follow me on Twitter, you will get lots of pictures like this because I am still learning my pollens, and so I end up having lots of questions. And I think that actually is an ACER. But from those pollen counts, you can start to put together a picture of what the plant community looked like in each of those different layers. And we can do that really well across the kind of lowlands of New England, and we understand that throughout the Holocene, we had this shifting mosaic of different natural communities moving in and out, lots of no analog communities, things that we think of as being really classic New England combinations, like beech and hemlock, didn't coexist until 4,000 years ago. And so when we think about how crazy everything was moving across the landscape over the last 15,000 years on our low elevations, it's kind of amazing that we just assume that the tops of our mountains looked the same that entire time. And so because I came into paleoecology as this like bright-eyed and bushy-tailed PhD student and had no idea what I was doing, I was like, let's get out there and actually core those high elevation ponds and find out. And so I went into Jacqueline's lab asking this question, are these communities, in this case Katahdin, post-glacial tundra relics? And so we had two field sites. The first one is Sargent Mountain Pond in Acadia National Park. Sargent Mountain is the second tallest mountain in Acadia, and this pond is pretty much right at tree line, and it had been cored in the past so that we knew it could be done, even though it's a little bit of a backcountry haul. And its claim to fame is that it's the oldest pond in Maine, meaning that as the Laurentide Glacier retreated, it was the first pond that was ice-free at the end of the Ice Age. And so we have a really nice long record. I'm still waiting on my carbon dates to come back, but other records have been dated from 15,000 years ago in that pond. And we think that we actually hit glacial clay at the end of our core. So we're hoping for a similarly long record. The other site that I looked at is Chimney Pond in Baxter State Park. So this is the pond on the back side of Katahdin. It's also pretty much right at tree line. And it is a really beautiful spot, but it is way into the backcountry there. So in both of these cases, I think that I really benefited from coming into this not really knowing what I was getting into. But I also had the advantage of, when I was in college, I worked for the Appalachian Mountain Club as a hut crew member, and so I really liked carrying heavy things to weird places in the woods. And I had lots of friends that wanted to do that too. So I'm going to show you some of our coring field work, starting with Sargent Mountain Pond. This is all the equipment that we needed to take up to our pond so that we could do our coring. We started this in September 2017, and we had things like PFDs and canoe oars. We had this long PVC pipe was full of the rods and our coring devices that we needed to get the sediment out. We had a lot of yellow or orange buckets, some empty tubes that we would be filling up with sediment. Katie and I are pulling a raft off the back of the truck while Jacqueline is supervising. And then we had to haul all of this stuff up the Deer Brook Trail, which is not a long trail, but it is pretty much all like that, really rocky. And we had these 2 giant plywood boards that would be the deck of our boat. And some of the maneuvering was a little bit tricky. And we lucked out that we worked with Acadia National Park, and Friends of Acadia basically gave us a volunteer group for the day. So this is a group of volunteers from a Sierra Club project that were spending a week in Acadia National Park working on different volunteer projects, and they came out in the field with us at the beginning of our project and at the end to help us haul equipment. And this is the group. They're really great. And Geneva, right here, took some of these photos. And then this is what the setup looked like once we had it on the lake, or on Sargent Mountain Pond. And we were anchored off right above the deepest point in the pond, and we were coring from that spot for about 2 days, and then we shifted over a little bit and cored a second hole so we could get overlapping cores. And so this is kind of the schematic of how it works. We had our nice raft in the middle. We set down our core. In the middle there's a rod, a metal rod that holds a piston in place until you get down to the depth that you want, which means you're not getting any sediment into that core until you get where you want. When you get to that depth, you pull up the piston and then you push down the core, and the piston at the top works like your thumb if you're holding the top of your straw, and the sediment is like your milkshake, so you can pull out a straw load of milkshake or a core load of sediment. And it looks like this. So from the middle of our boat, remember we had 2 plywood pieces, which meant that there was kind of a hole in the middle of our raft. We set down the core and we had metal rods. And there was a day that it was just me and Jacqueline on the water, and we called this coring Margaret Davis style. Margaret Davis was like the pioneer of sediment coring across New England. And it used to be just her and a single grad student would do these like incredible, like 14-hour days of just pulling up sediment. And I don't know how she did it, because after that day I needed to like sleep for a week. And you pull up the core and a lot of water comes out. And it looks like this as you get it out. You can see all of our sediment in this clear polycarbonate tube. The park uniform behind us is Abe Miller-Rushing. He came out coring with us for a day. He's the science coordinator at Acadia National Park. And that was me and the very first core that we pulled out from Sargent Mountain Pond. It was very exciting to finally, after all this planning and work, to just get all of the equipment up there to actually see the sediment right there in front of us. And once you had your core collected on the raft, you needed to get it back to the mainland. So we had a little red kayak that we would send people in and out with the cores. And you had to hold them up so that you didn't mess up your layers as you were paddling them out and then hiking them out. And so this was one of our first day's hauls of cores that we pulled out and put in the back of a pickup truck upright. And that was a great experience in Acadia National Park. The park let us basically leave our raft equipment out there on Sargent Mountain Pond for about a week, and we just hiked in and out each day. We had hikers walking past us on the trail, and a surprising number of them knew exactly what we were doing. So I'm not sure if it's just that like every pond in New England has been cored so well that like the hiking community is just like totally numb to it, but a lot of people understood fallen cores. And so for Chimney Pond, we were working with Baxter State Park, and initially when I was trying to get a research permit with Baxter, I was planning to do the same thing that we had done in Sargent Mountain Pond, hike in the summer with our boat, set it up, and camp there for the week. And the Baxter Rangers got back to me and they said, well, we're actually trying to do a bunch of trail work on the trail up to Chimney Pond, and we want our trail crews to be able to use the crew cabin up there. But what if we snowmobiled you in the winter and you did your coring on the ice? And that was just an incredible offer, because the amount of equipment that you have to get in to Chimney Pond, and in the winter it's a much longer haul because they close off the road, meant that snowmobiling made it just incredibly easy. And so we loaded up all of our stuff into the back of the snowmobiles, And we also got to ride in the snowmobiles for part of it, and then we skied up the last like 3 miles on a beautiful bluebird day. And these are all folks from Jacqueline's lab. This is her PhD student, Dulcinea, who just graduated last fall. Karen James, who is a researcher from Bar Harbor that's now working with our lab on some eDNA stuff. I'm in the purple jacket, and then Kit Hamley, who's a current PhD student. And so we had a wonderful week working in Baxter State Park in March of 2018. The only, I would say, like real challenge of setting up this field work was we were so dependent on the snowpack, and the winter of 2018 was just a slow build. There was a lot of rain early in the season, and so the snowmobiles weren't actually going up to Chimney Pond for a while. And when we had initially put our plan together, we thought that we were going to be doing field work at the beginning of February, and we didn't do it until the second week of March, just because it took that long to get enough snow. And that was a little bit troubling because in order to do coring on the ice, you have to actually get through the ice. And so this is us going out onto the pond. And so we were nervous that we would be hand augering through many feet of ice. And I had never been ice fishing before, but we watched a lot of ice fishing YouTube videos to get prepared for this field work. And the one tip I would recommend is if you bring PAM spray, like cooking spray, and you spray your ice auger, it actually moves through the ice pretty well. And we were lucky that there were only about 3 feet of ice here, so it took us not very long to get an open hole. And we, again, opened one hole and then moved over a little bit a couple days later and did a second hole. And this is what it looked like when we were coring through the ice. So again, we had the rod connected down to the core through our ice hole, and then we pulled up cores a little bit at a time. And one of the really nice benefits of working at Chimney Pond in Baxter is they were putting us up in this crew cabin, which was incredibly cushy, because in the middle of the day, you could just take your core into the cabin and do some of your work by the wood stove. It was really lovely. And so this is us doing some core description on one of the cores that we pulled out. And so at the end of our field work, we still had a ton of sediment that we had to process and get ready to do our actual analysis on. And so in order to get a core that looks like this, I did take all of these cores that were nicely wrapped up out to University of Minnesota, where they have the National Lacustrine, what's the rest of the black core stand for? National Lacustrine Core something facility, where they do all these initial core descriptions. So they x-ray your core and take high-resolution photographs of your core, and then help you cut your cores in half. So you're basically working on these long half cylinders, and starting to do things like pulling out macrofossils along the top, so things that we could carbon date from our core. And I should point out that this picture along the side of my standard core, it came from the high resolution photography that they did at the LAC core facility. And then I brought my cores back to the University of Maine, and we started cutting them into little slices and sampling them for pollen. And so you would spend a couple days at a time basically slicing each core into half-centimeter cookies. And then each half-centimeter cookie got a little baggie in a nice box. And then every 8 half-centimeters, I took a 1 cc sample for pollen processing, and that went into the test tubes. And so ultimately I made pollen slides from those. But each of the baggies over there gets put through a sieve so that we can look for macrofossils. And I worked with one student, Cass Carroll, last year, who wrote her senior capstone paper at University of Maine on the macrofossils in the bottom 2 meters of our 4-meter long core from Sargent Mountain Pond. And she collected some really beautiful macrofossils. This is a Poaceae. B and D are betula. I think B is a seed and D is from a catkin. And then this is a spruce needle. And so she kept track of which macrofossils she saw in which layer. We were looking for evidence of either changing plant communities or consistent plant communities. And over those bottom 2 meters, she found pretty consistent evidence of spruce, fir, and birch macrofossils. So it'll be interesting to match up my pollen records with that. And pollen is, as you know, pretty light, and so it can travel from much farther away. Whereas these macrofossils were things that were growing right there at Sergeant Mountain Pond and fell in from right next to the pond. So we'll see how the local macrofossils match up to a more, a broader kind of pollen shed of the rest of the mountain. And so, getting back to my original question where I was asking, are these communities post-glacial tundra relics? My initial hypotheses were that we would expect these diverging evidence for transient forest communities. So I expected that Sergeant Mountain Pond, which is this low elevation site in Acadia National Park, we'd see lots of evidence of trees coming in and out in the record. And at Chimney Pond on Katahdin, which is a higher elevation site right next to the largest alpine area in the state of Maine, there would just be really consistent open plant community evidence. So those are my initial hypotheses, and I haven't analyzed enough pollen or macrofossils to prove either one of them right or wrong, but I'm excited to find out. And I think what I ultimately want to do is not really ask about, is that story about tundra relics right, but can we use this information to determine how vulnerable these communities are to anthropogenic climate change? So when we look at something like this really recent publication that looked at incorporating climate change refugia into climate adaptation in Acadia National Park, which all of the managers in Acadia are reading, they're trying to identify these climate change refugia, these areas that are buffered from contemporary climate change and enable persistence of physical, ecological, and socio-cultural resources. Well, they're doing this from a really sophisticated modeling perspective, looking at climate models. But what if we look at this from a different angle and look at it from a paleo perspective and are able to identify locations that have over the Holocene served as climate change refugia? And how might those inform those models for identifying climate change refugia going forward? So ultimately, I'm hoping that the results from this work can be really directly applied into conservation in a way that I think a lot of paleo research doesn't yet get into, even when it's generally could be applied on the ground, doesn't match up with management directly. So I just wanted to end with one of my favorite photos from our fieldwork in Baxter State Park. And if you ever get a chance to do winter coring, I highly recommend it. It was just the most incredible experience. And I would say that working in Jacqueline's lab has given me this new perspective on all of these plant communities, whereas as a PhD student, I used to walk through Acadia and try to imagine what the landscape looked like 100 years ago when Edward Rand and his Harvard friends were doing their original floristic inventories. And I always thought when I was a PhD student that I was really deep for thinking that way. I'm feeling, oh, I can imagine the landscape 100 years ago. But working in a paleo lab and coming to a paleo lab from an ecological perspective has given me so much respect for the amount of work that geologists and paleoecologists do, because they're able to hold these huge time spans in their head and look at temporal variation in a way that I think most ecologists can only imagine spatial variation. So I wish that Jacqueline was here to talk to you today. I'm excited that she'll come down hopefully in the spring. But this is just a little piece of what we've been doing in her lab over the last 2 years. And I'm happy to answer any questions about it. This is I'm sure this slide gives out a lot of people. I don't even think Karen James is on it because I wrote it about an hour ago. But I had really wonderful support both at University of Maine, in Acadia National Park, and in Baxter State Park. And I did this work as part of a David H. Smith Conservation Research Fellowship and Second Century Stewardship Fellowship from the Schoodic Institute. So thank you, everyone. Yeah.
For ice cores, you have rather accurate yearly data. Do you have yearly markers with these sediment cores, or is it really just carbon dating?
It's really just carbon dating. So our sites are not laminated with yearly laminations. So there are some really beautiful sites actually in Minnesota when I was doing some training where we got to see cores where you can actually see the yearly sediments laid down. and do really beautiful dating. But I'm waiting on some carbon dates to do age depth models, which are fairly accurate, but definitely not as nice as having those yearly dates. Yeah.
Is there, does the climate data for that area tell you whether in the Holocene, when it was warmer, did it ever exceed where we are now or where we're about to go? Or are there points in the past that were actually warmer than today?
Yeah, so for the region coming out of the Younger Dryas, there was a rapid warming period, which seems to have been about as warm as today. But we don't have climate proxies for these two locations, and one of the things that I'd like to do now that we actually have collected the cores is reach out to some folks that do climate reconstruction with non-pollen things. And so we've archived half of all of our core material at LACOR, and it would be great to get really localized climate proxies for both places. Yeah.
How far back did you get, or can you tell yet?
We can't tell yet. In Acadia, we got to what looked like glacial clay. So the whole core was this like really dark, organic, like cake batter type sediment, and then at the very bottom, it turned to gray clay. And so we think that was glacial clay. But we're waiting on our carbon dates to come in. We just sent those out over the summer. In Chimney Pond, which was cored for geological reasons in the 70s, they were only able to get back to 8,000 years, and I don't know if that's an idiosyncratic thing of that particular core, or they had this theory that the whole basin basically just like washed out 8,000 years ago. And so our core might not get back farther than 8,000 years. There just might not be sediment that far down. In Chimney Pond, I'm pretty sure that we hit the bottom. Like I think that we hit bedrock when we got to the very bottom. We actually cored through like a stick that was about this thick at the bottom of a core that we thought, like when we were getting that core, we thought that was the bottom. We pulled it out, realized that we had a stick, and went back for more, and we were able to get a little bit deeper, but then hit something. Yeah.
I'm wondering about adaptation to different climatic regimes. These studies are really cool for thinking about migration and where the plants go, but it's hard to account for adaptation.
Yeah.
So I'm wondering, do you find the same vegetation assemblages, though, that you see today in the core? So all the same tree species co-occurring, just like
that's
modern?
That's what it seems like from the macrofossils, which again is just the bottom half of one core at Sargent Mountain. We haven't done the macrofossils from the top of Sargent or from Baxter. And my pollen counting has gone, I think I'm at 40 centimeters from the bottom now. So like I'm much slower at pollen than my students are at macrofossils. And so I don't know yet, but it would be really interesting to find out. And then I think just from my perspective working with these plants as a PhD student, I looked at a lot of, almost all the species that I looked at shifted their phenology with warming temperatures in the spring. And so they do seem to have some plasticity in how they're responding to temperature changes. And I would really love to tie the paleo work back to some more direct studies of, especially the Alpine areas, how those Alpine plants are responding not just in phenology, but how they might be adapting in other ways as well.
Yeah, cool.
Yeah. Yeah.
I'm wondering if there are any sites further south where you might have longer histories, take you back for a very time in the southern Appalachians. There's a place called Ripshin Bog below Roan Mountain at around 3,800 feet, place like that. Pollen cores that might tell you about what's happened on the upper elevations of Roan.
Yeah, there have been a couple cores from the southeast, but none that I know of from high elevation in the southeast.
Yeah, it's hard to find them something real high up.
Yeah. Yeah, and then you have to find sediment that you can actually get through.
But even Chimney Pond is going to be pretty low,
Yeah.
relative to the top.
Exactly. Yeah, and there is a pond on Tumbledown Mountain that's like in western Maine. That's shorter. The mountain itself is smaller than Katahdin, but the pond is higher than Chimney Pond. And so if I can figure out a way to get corn from Tumbledown Mountain's pond, it would be really interesting to see what's happening at the highest elevation site. Yeah.
What was your weather like at Chimney Pond?
Our weather was amazing. We had a beautiful ski-in day, and then we had built in one storm day, and so we worked for the afternoon that we skied in. One day there was a nor'easter. We stayed inside and ate, and then skied a little bit, and then we worked on another full day after the nor'easter, a morning, and skied out.
What were daytime temps?
They really actually weren't that bad. I think we were about 25 most of the time. And we had that cabin, and so even the day after the nor'easter was the coldest day, and even working that full day, we would come in and warm our hands up and then not have a problem getting back out there. It was mostly that we really lucked out that we were there in March because our days were longer than they would have been if we were there in February. And so we were able to like really take advantage of having nice weather when we had it, so that we didn't feel bad about missing a whole day when it was snowing.
Did you find coring through the ice a lot faster and easier than coring from a boat?
It was a lot logistically easier once we had the hole augured, because we could just spread out all of our stuff everywhere, and you could have people walking in and out at different times. With the boat, we were constantly like, the kayak was in the wrong place, or we had the wrong person, or we had like left something back on shore. And so it was really nice to like just take our sleds out onto the ice, open up a tarp, and just like throw everything down. So I liked that a lot. I can imagine that winter coring, if you didn't have a cabin, would be a lot harder and a lot less pleasant. But for what we had, it was just amazing. Yeah.
How accessible or how much can you look at other cores and other ponds and compare, you know, what you're going to find at these two ponds to other places? I don't know how accessible that data is.
It depends. There's a lot of places, or there's a lot of cores that still exist that are archived, and so like that archival material is available. There are a lot of the slides themselves that are available. So before we did this work, there was that core from Lakes of the Clouds, and then there was a little bit of work done at in the Adirondacks across like an elevation gradient by Steve Jackson. And Steve Jackson's pollen slides are now at the Harvard Forest, and so if I wanted to like actually look at his pollen slides and compare what was happening in the Adirondacks to what we find at these sites, that would be possible. In a lot of cases, it's just going back into the literature and looking at their pollen diagrams. And so, especially like for the case of Lakes of the Clouds, they were looking, I am looking at pollen every 4 centimeters. At Lakes of the Clouds, they looked at pollen every 10 to 20 centimeters, so the temporal resolution was much coarser. And again, because I came into this with no idea what I was getting myself into, I was like, yeah, sure, I would love to look at 100 pollen slides for one core, which I'm kind of regretting right now. But it means that if those pollen slides don't exist and that archival material doesn't exist, I can't do that same like temporal resolution comparison, because if they only have every 10 to 20 centimeters, and I'm trying to fill in the gaps, it's hard to make that comparison. So it's really important to archive your material and get metadata on everything. Yeah.
How well preserved are you finding the pollen in your cores? As I recall, there's a lot of variation. Is that good
Yes,
stuff?
yeah, these cores, I mean, the Sargent core in particular, I've been working from the bottom up, and it's just really nice. And so I've heard from Jacqueline that I really lucked out on my pollen, which is nice to hear, but also frustrating because it's still hard to identify. Yeah,
Are there resources out there where you can take a shot and send it off to someone in Wisconsin who's a real hot shot and get an ID?
So there's a Global Pollen Project website that collects a bunch of photographs from around the world. It's really European focused. There's a online community of people in mostly based in the U.S. that share pictures back and forth. And then our lab, right now, Dulcinea and I are both working on pollen from different projects. She cored in Alaska a couple of years ago. And so we, within our lab, have an online resource with the two of us where we send photos between each other and to Jacqueline, and she helps us with our identification. And I'm not on campus very much this fall, but when I am on campus, and when I'm not on campus, they do it anyway, they have a pollen clinic every Wednesday morning where they basically spend like 3 hours in the microscope room together, where sometimes it's just everyone working quietly, and sometimes it's just like lengthy discussions about like, what do you think this is? And we have all these reference slide materials, so I have like a whole box of reference slides. So I have a slide for birch, and I have a slide for alder, and I have a slide for spruce and fir. And so going back through these reference slides is really helpful. But then sometimes you'll have a reference slide that somebody collected that says it's spruce, but it's also full of birch pollen because they just like collected at a time when there was just birch pollen in the air. So it depends. But it's been really great to have other humans working on the same type of struggles and being able to reach them through online communities is great. And like that has been a huge part of why I joined Twitter, was to find other scientists that were struggling with the same things I was, which at the time was I wanted to spy on people in Maine and make sure their plants weren't blooming yet, because I was doing fieldwork in Maine, but a student at BU. And now it's like I want to reach out to other people who are also struggling through the pollen and share silly pictures, like my picture of the birch and the beech with a question mark over. I really do think it's an acer. We'll find out. Yeah.
Are you able to tell the difference between species within a genus?
It depends. You can't for birch, or I can't for birch. There's some maples that you can tell apart. There's some spruce, so red and black spruce you can't tell apart. There's another one that you can. Oh, pine. You can tell white pine from jack pine based on size mostly. But for a lot of it, it's just the genus. And so that's another difference between working with macrofossils and working with pollen. Sometimes with macrofossils, you can get down to a higher resolution of the taxa. And with pollen, you're just kind of wondering, is this, a heartleaf paper birch, or is this a paper birch that could be like anywhere on the mountain? Yeah.
What sort of diversity do you see in those, like an individual cross-section or cookie? Like, are you encountering 10 or 20 different types of pollen, or 5, or?
So each slide that I've looked at so far has had between like half dozen in the early, especially like the early levels, which were a lot of just blanks, to probably 20, maybe 25 in the really busy ones. There's also a lot of, like, you get aquatic stuff in there that you're counting even though you know it's not actually helping you understand what's happening around the pond. And so, for example, my undergraduate just found like hundreds of macrofossils of like algae stuff. And it's like, oh, well, I'm going to count it and sort it, but it's not going to tell me what's actually happening here. Some other scientists might be interested in what's happening in the algae populations, but like we don't use it for our study. And she found a lot of hit or miss. Like some of her samples had nothing in them, and some of her samples had lots of fossils in them, and it didn't seem to depend on depth for her. Whereas I found it very much depends on depth. Where I started at the very bottom of my core, and my first 5 slides, it was like, there's nothing. Here's one birch, awesome. But now I'm getting into more diverse communities with lots of pollen.
Are there charcoal bits?
I'm not counting charcoal bits, but
Are you seeing them?
I think that I would be seeing them if I was looking for them, because the other core, or the earlier core from Sargent, was worked up for charcoal, and they found like a series of different fire regimes. So I don't know, because I don't study charcoal, so I haven't like trained on it, but I haven't noticed it. Yeah.
I'm curious. So you have like 100 baggies of dirt now, pretty much? Or
I have more than that. So I have 4 meters, and I have every half centimeter. So I have 800 baggies and 100 slides.
It's just, I assume they're cataloged, and if somebody does want to come back and look at charcoal,
Yep.
or like as you refine your search, there's gonna be more questions. Can you go back to it pretty readily?
Yeah, so the baggies you can't go back to once they've been sorted for macrofossils, but the baggies are only half of the core, so the archive half of the core is still in Minnesota in a giant, like, refrigerated room there. And it can be accessible for anyone who's interested in various questions, so they just have to, like, reach out to the facility, they get my permission, and they can use it for anything. So we've already started working with a group in Canada that's looking at dissolved oxygen content in the top couple centimeters. And I don't really understand their methods of like how they do things with a freeze dryer to figure out dissolved oxygen, but they've gotten to pull some samples from the same core.
So I wonder whether it would be possible to, say, take a photo of your corner slide and work with somebody, say machine learning or something. They
I
can
would love that.
cluster
So
different type and then you can.
there's a couple issues that make it especially difficult for machine learning. The first is that you're looking at a 3D image in 2 dimensions because you're looking at a microscope slide but trying to get the pollen. The second is that you're working with pollen that's 10,000 years old, and so it's slightly degraded. So even though my pollen is pretty good, it's not as good as fresh pollen. So if you train a machine learning algorithm on fresh pollen, it would just take a lot of effort to get it to learn all the different 2-dimensional shapes that you could get from one 3-dimensional piece of pollen. And then also to be able to figure it out if it's slightly degraded. And then also, the slides are full of lots of things that are not pollen. So the first thing that I had to learn, which I basically spent this like past summer on, was how to identify what is pollen and what is not pollen. Like not like, is this a birch, but like, is this a piece of pollen or is this just debris that I don't have to worry about? And so that, I think those steps of training make it really difficult. I've heard of lots of different efforts to work on that problem. I think a lot of paleontologists would love it. And it's been actually really reassuring for me to hear from paleontologists who are like really well established in their careers talk about how difficult it still is for them working with pollen. But that makes me think it would also be difficult for a robot. Yeah.
So what's the chance of, when you're making the slides, contamination from pollen just floating around in the air?
There is a chance of it, so we do it in the lab. But because I'm not working with like eDNA, I'm not like in a clean suit. All of the pollen processing happens in a lab, like under a hood and stuff with acid baths. So yeah, it's definitely possible. For each slide, I'm counting between 300 and 500 individual grains. So if a modern pollen got in there, it would probably be swamped out statistically anyway. Yeah.
What is that, can you give us a quick overview of how the pollen slides are made? Do they freeze the core and microslice it or something?
So the core is refrigerated in a cold room. I took out each core, sliced every ½ centimeter into those baggies. Every 8 baggies, I took one, it was like this little square. It kind of looks like it would make a dye. Like you just you stuff sediment into this little square cube thing and drop that cube into a test tube. And then I actually learned how to do this with Jacqueline, but then I was pregnant, so I was not allowed to actually do the acid baths myself, so we set them out. But basically, pollen is so resilient that you just put the sediment through a ton of acid baths, and everything else burns away, and all you're left with is pollen. And so you get rid of the other organic material, you get rid of the other calcites. And so you end up with a little tiny vial that's full of pollen, and then you take a little dropper of that vial and some silica gel, and you put it onto a glass slide, put on your little slide cover, and then you use Sally Hansen hard as nails and dot each of the corners and draw the square to hold your pollen slide together and let it dry. And usually if you don't dilute that droplet, or at least for my samples, if you don't dilute that droplet too much, you can get all 300 grains that you need to count onto a single slide. So far I've there's 2 slides that I need to remake really badly right now. One is because I got it really bubbly. The other one is because it just didn't have enough pollen on that one slide. But the process itself, the acid bath process itself is really interesting and maybe not fun, but like I found it, because I was a field ecologist, interesting to do some lab chemistry stuff when I became a postdoc. And then the pollen counting is monotonous, and I have a little microscope at my house, and I sit down for an hour every morning and try to work through a slide, or half a slide, or sometimes one corner of a slide. Yeah.
Well, thank you very much
Thank
for coming
you.
out on short notice.
No problem.